to, becoming an addiction neuroscientist and wanting to make discoveries that have an impact on our field started in Jamaica. But I was coming back from a conference on the flight, and I ended up sitting next to someone I didn't really know that well at that time, Diana Dow Edwards. But one thing my mom, sorry, I'm getting a little choked. But one thing my mom did, I think the foundation is character. I forgot to really say it. It's embodied here. It's international. And so that's the thing, you belong to a very special group. Einstein became like my hero. And so people like, this young Black girl, what she liked, Einstein. And that's cemented to me that, yes, I have a scientist heart, and there's something there. I have no idea, like I said, where it came from, that science was going to be the path. So that was my beginning and how I started, towards my love of addiction neuroscience. So I was like, okay, here I come. I'm going to change the world of addiction neuroscience.
And today is one of those role models. I have been following Yasmin for a long time.
Question is, can the neuroscience knowledge advance novel treatments? And so that's also, a huge part of the work that.
So, hello everyone, welcome to the 12th and the last talk of ISAM-NIG talk series in Love with Addiction Neuroscience. Thank you very much. So I'm now going to unshare my screen. And Dr. Baldacchino, you may want to start the introduction of the speaker. Thank you.
Thank you, Parni, and good morning, good evening, and good night for some people. Mm-hmm. And I'm really, really glad to be here, and I'm even more glad to chair this meeting because I was invited to chair the first meeting of this series, which was 12 months ago. My name is Alex Baldacchino. For people who don't know me, I'm the International Society of Addiction Medicine president, but I'm also a clinician and an academic working in the United Kingdom. But the ISAM community for me is a platform where we can share common themes. But most important of all, and this is in discussion when Hamid and other members of the Neuroscience Interest Group we're discussing the prospect of organizing this series, is the notion that we're all human beings, we're all, have our strengths and weaknesses. But one important thing is that we need to encourage the younger generation by having not only honest and decent role models, but also exceptionally inspiring role models. And today is one of those role models. I have been following Yasmin for a long time, academically, I mean. But I'm really glad that now she's going to give us a talk. I'm going to give you a brief bio of Yasmin, and then I'll hand over to Yasmin to give us the presentation. Dr. Yasmin Hurd is Professor of Psychiatry and Neuroscience at the Icahn School of Medicine in New York, USA. She's an internationally renowned neuroscientist whose translational research examines the neurobiology of drug abuse and related psychiatric disorders. She's highly published in the field and leads a team of investigators in molecular biology, behavioral neuropharmacology, genetics, and neuroimaging to study the human brain as well as translational animal models. Dr. Hurd is also director for the Addiction Institute within the Mount Sinai Behavioral Health System, which covers one of the largest addiction populations in the US, providing clinical care supported by science-based evidence and advanced state-of-the-art research. I'll hand over to Yasmin. Thank you.
Thank you so much, Alex, and for the honor of closing this first year series, which I think has been very successful. I'm trying to just move it's not that I don't want to see you all, but I just want to be able to move you down here. Yeah. So as I said, thanks very much. I mentioned that. Hello? Okay.
Yes.
Yeah, so I mentioned earlier that I'm on vacation, and I had meant to put together a couple of things regarding my love with addiction neuroscience. And unfortunately, I lost a few of them along this way. But I recently gave a talk on Black and Neuro, which I think is a good foundation for this journey that I've been on. And one of the things for me, as you the posters mentioned, that my road to becoming an addiction neuroscientist and wanting to make discoveries that have an impact on our field started in Jamaica. And many people, I think the last time that someone asked, can we get a scientist, representing Jamaica? And I don't know if I still qualify for that, but definitely a huge foundation for who I am, the way in which I think I conduct my research. I think, persevering through this path where there weren't that many role models in the sense of, people who looked like me necessarily. And it all started in Jamaica. So it is possible to come from a very small island and to do, I think, strong research and respected, by your colleagues. But a huge part of that stems from my mom. Well, my dad also, but I'm going to focus on my mom in large part. So my family, or academics or education, was huge in the sense of, doesn't matter what you do. My mom always used to say, make sure you get a piece of paper. So everybody thought, when I was younger, I was extremely curious. I would always try, to learn as much as possible. And so everybody thought I was going to be a teacher. But I loved science, and I have no clue where that love of science came from, because nobody in my family are scientists or physicians. My mom loved the English language. Books were her life, and books were my life, but mainly actually science books. My mom, to tell you in terms of the English language, my mom would correct my emails that I sent to her. So even though it doesn't matter what age, she would do that. So everybody thought, okay, teacher or doctor that I was going to be. And for doctor in Jamaica, they think of physician. No one, as I said, thought about scientist when they were talking to me. But because I did talk a lot and I was always curious, people thought, okay, she could also be a lawyer. But one of the things for me in terms of Einstein became like my hero. And so people like, this young Black girl, that she liked Einstein. And that cemented to me that, yes, I have a scientist heart, and there's something there. I have no idea, like I said, where it came from, that science was going to be the path. So my path is based on the fact that I moved from Jamaica to the US, and in particular New York. Unfortunately, my parents divorced, and but I thought it was a great win for me, because I got to go to live in my favorite city in the world, New York. And there, I loved sports as well. I was on a track and field team. I was crazy when I look back that I was a gymnast, specifically my specialty was the beam. But what won out always was science. So no matter what I did, I kept on coming back to this aspect that's what I loved. When I went to college, I was also a little strange in the sense that I went to SUNY Binghamton, which is upstate New York, state college. And at that time, there was no neuroscience, not just at Binghamton, anywhere. There was not a neuroscience specialty. But again, I was weird in that I created my degree was actually a specially created major, and I combined biochemistry and behavior, which basically was neuroscience, and I didn't even know it, because I was just really interested in chemistry and also the chemistry behind the behavior. And I was working to make money because I didn't have much money, and was my work study was taking care of the animals in the vivarium. And this amazing professor there, Peter Donovic, who I have here circled, Peter Donovic, I want to make sure I keep on time. Peter Donovic, I was taking care of animals for all of the psychology researchers, but I kept on asking questions. I guess that's the commonality throughout my life, from the kid to now even in school. And he was like, "You're asking even better questions than my postdocs. Would you like to work and do research in the lab?" And I said I couldn't because I needed to make money. And he organized for me to do research and still get paid. So he started really my more academic understanding of what it meant to be a scientist, and gave me an opportunity. And I found my first paper, and where I looked at the effects of parasite actually on behavior and brain. And Peter Donovic, he just like completely believed in me. And so when someone believes in you, you actually, work towards making sure you live up to that. So I always appreciated it. In fact, it's crazy that life can be a full circle. Nearly 25 years or so after I graduated, I was the graduation speaker for at Binghamton University, and with Peter Donovic there and a lot of my close friends who watched my journey along the way. But the journey into addiction neuroscience actually came from my venturing to yet another country, and this time Sweden, and in particular the Karolinska Institute. And the Karolinska Institute was renowned because that's where neuroscience was really beginning to blossom. And in particular, Professor Urban Ungerstedt, who I had met at a conference, and again, I was asking many questions, and he was like, would you like to work on this project? And it was a project to measure neurotransmitters, in particular catecholamines, in the living brain. And he was trying to develop a technique called in vivo microdialysis. But Dr. Ungerstedt, he, for me, was like my god of science, because he was part of the Karolinska scientists who had mapped the catecholamine systems in the brain. He developed the animal model, the 6-hydroxydopamine lesion animal model for Parkinson's disease. And as I said, and he was trying to develop an in vivo system to measure dopamine in the living brain. And dopamine was such a critical transmitter for a number of, especially at that time, I was interested in neurodegenerative disorders like Parkinson's disease. And so for me, it was a really great opportunity to be at the Karolinska. And one thing about being at the Karolinska, and I always tell people this, for me, all throughout my life, and especially in the US, being a Black girl who likes science, there's questioning, and nobody expects you to succeed. But at the Karolinska in Sweden, it was actually the opposite. They were like, "Oh, you're American, so you must be the best." So therefore, they set this high bar that I must, be better than everybody else. And that was actually refreshing, and actually gave me the confidence that, yes, I can do this. So, as I said, the microdialysis technique definitely changed my life, that moved me towards addiction neuroscience. And the reason is that the in vivo microdialysis is that you insert a semi-permeable membrane into, brain regions of interest, and chemicals flow in and out of the microdialysis solution, CSF that's flowing through it. And so you can then, by HPLC or other techniques, measure what, in a time-related manner, the neurochemicals that are changing. But because we were studying the dopaminergic system, for aspects of looking at dopamine function relevant to Parkinson's disease and other neurodegenerative disorders, we actually started looking at drugs such as amphetamine, cocaine, these psychostimulants, whose mechanism of action were targeted to elevating dopamine in the synapse. And we were able to see, using the microdialysis, that when you gave these animals cocaine, you could see this rapid effect on increasing dopamine in the brain region. Here, it's the nucleus accumbens. And the thing that was exciting for me was that the time course, and being able to actually study the time course of in vivo neurochemical fluctuations of dopamine, we could see that that time course matched the time course that was being observed in a recent technique that was in terms of in vivo neuroimaging, PET imaging. Here it's Nora Volkov and her, who's now the current head of NIDA, but at that time at the Brookhaven National Laboratories, looking at where the impact of cocaine on the brain and looking at C11 cocaine. And they could see that the time course really matched the high that people felt, and it matched what I had seen in using our in vivo microdialysis technique. So again, that was exciting, but I wanted to see, was it relevant to the actual self-administration of cocaine? So in combining in vivo microdialysis in the animals that were self-administering the drug, we could see indeed that the time course of dopamine fluctuation actually correlated very well with the self-administration of cocaine itself, which now, we're like, duh, it should have, but it was great to be able to see that and get, like I said, the time course. But the thing that really got me and turned me into wanting to look more at drugs was the fact that the power of these drugs in changing behavior. It was incredible when I saw how strong the behavioral changes were in the animals with the self-administration of these drugs. And indeed, I was able to even show that, animals would maintain a certain level of cocaine if they were given free access, given, certain level of cocaine in their brain. Here, looking at the nucleus accumbens and amygdala, also that each of these animals that was found rewarding. So that was my beginning and how I started, towards my love of addiction neuroscience. So I was like, okay, here I come. I'm going to change the world of addiction neuroscience. And then I had, a number of my close friends, my best friends in college, they stopped me and they're like, Yasmin, but what do you really know about drugs? Because they then reminded me that in college, I didn't even drink alcohol before I came to college. And, people always say, oh, you're from Jamaica, so you must have, smoked marijuana. And I had never done that. And, so they were like, don't you need to have had experience with the drugs in order to study it? And I was like, no. But they were my best friends then. They're still my best friends now, how many years later, because they were like, okay, we're behind you 100%. Whatever you want to do, we're supporting you. And they've always done that. So for me, the journey to then follow the path to addiction neuroscience came at the NIH, or continued, I should say, at the NIH. And there I did my postdoctoral training and took my first independent steps in running my own research group. But for me, something that I think, which framed the foundation of the research that I do is that I wanted to make sure that a lot of the work that I was doing had direct impact to the human condition. And especially at that time in DC, crack cocaine was, really a scourge, I could say, on the communities. Many people were dying, and it was really important for me, and that's why, why am I committed to addiction neuroscience? It's about addiction and the people. And just, it wasn't just that it was changing behavior that I could see so clearly in the animals. It was that it had such a huge impact on people's lives. Every day you could hear about these kids and adults who were dying, and they were, it was just horrible. But so for me, the question was, what could I know that we couldn't study, use the in vivo microanalysis in the brains of people. We actually did one or two in people that had Parkinson's, but in terms of substance use disorder, and for me, it was, looking at molecular changes in the brain. And in following the dopamine system of what I saw that was altered, the question was, could we see molecular changes relevant to the self-administration of cocaine? And here, so at my postdoc, I then switched from being mainly in vivo neuropharmacologist or biochemist and started, like I said, learning molecular biology so that we could look at gene expression changes. For example, this is prodynorphin in the rats that had self-administered cocaine, and we could see this, really strong upregulation of the dynorphin gene in our animal models. But the direct relevance to the human condition was also a critical part for what I knew long-term. My goal in the ultimate aspect of my path in addiction neuroscience was to see if what we've learned about the brain can help inform treatment. And so could I study the human brain? And that was, again, where a lot of barriers were put up that, it's not possible. Molecular changes that you see in the animal brain, you know everything about the animal's history, and the brains are so pristine with animal models, while the human brain, you don't know when the person died. Molecular mechanisms or molecular changes are all, dysregulated at the time of death. And I'm like, but, we studied the animals after death. So it took a while to convince, actually, a lot of people that I could and I wanted to study the human brain and leverage what I had been studying in the animal brain on a molecular level to the human brain. And indeed, we were able to see, I see some of this didn't, but in any case, we were able to see significant changes in, for example, here, the dopamine transporter in heroin users in on the gene expression level in the VTA, the ventral tegmental area, and we could also see it at the protein level in the nucleus accumbens. However, for me, in trying to follow what was the effect of changing this in vivo dopaminergic regulation on a molecular level, it came back to the dopamine receptors in terms of if dopamine was released in the nucleus accumbens and was so critical for reward and addiction itself, then, as the hypothesis really was at that time, then the cells that are reading this dopaminergic tone and transmitting this tone to mediate behaviors, what do they look like? And that's when, for me, the molecular biology was key, because then we could look at the cells that are expressing the dopamine receptor genes and protein, but in particular, we can start looking at pathways. And so, for example, we know in the striatum that the heterogeneous populations of cells that make up the different pathways. And for example, those cells that express the dopamine D2 gene are part of the indirect pathway, and they express the proenkephalin gene. And those that go back to the nigra or the VTA area that express the dopamine D1 receptor expresses the prodynorphin gene. And so we could see in studying cocaine users that we actually replicated what I had seen in animal models, that there was this increase in dynorphin. But something that was important was that we actually saw something different for the proenkephalin. In animal models, or a rat, I should say, we never really saw that there were significant changes on enkephalin. But in the human cocaine users, we always saw this imbalance between dynorphin and enkephalin that we later on were able to replicate in a monkey model, so that there was a rodent and primate difference here. So for me, it really helped to set the foundation for what kind of neuroscience research I wanted to conduct in relation to addiction. And actually, I then, between Sweden and the US, going back and forth, I developed my research program as an assistant professor, associate professor, and then and I'm going to give you some of the neurobiological, I guess, my path to developing my addiction neuroscience program. And for many people, they think, okay, it's a lot. But for me, that's how my brain works. And my brain worked in that way because another thing that fascinated me about addiction, and I saw this, on many levels, as all of you know, addiction is a complex disorder. And it's not everybody who will develop an addiction disorder. And the question is, why not? We know, of course, that the drug itself, right? But we know that genetics plays a role. We know that behavioral traits and psychiatric comorbidity plays a role. The developing brain in terms of the time period of exposure is important, as well as the environment. And that has set the foundation for the path of my career in appreciating the complexity of it, and delving into each of these in a little way, but using the human brain in large part to help guide some of that. And some of it was fortuitous in the context, or serendipitous perhaps, where, for example, I'll show, in terms of some of our studies with genetics. So one of the things that was really critical for me was when we, so I developed a human brain bank collection, and with my colleague, Eva Keller. And one of the things that was, Eva Keller was at that time heading the forensic department in Budapest. And I was in Sweden, and I wanted to develop, as I said, this human brain bank collection. And I had reached out across Europe to many different pathologists to see if we could combine our efforts. And Eva was the one who was consistent, and we became great friends, but important, great colleagues. And for me, what was important about this brain bank collection, and it taught me something even back then, was we were both in countries where, not unlike today, drugs were not very prevalent. So people who were, really engaged in substance use and became addicted to drugs, there was something that they had to really go out and find it, because as I said, it wasn't that it was as prevalent, and definitely not like the United States where drugs were, quite common. And even back then, it was clear that opioids obviously were of the greatest risk because of mortality. Our brain bank collection ended up consisting predominantly of opiate users. And that's when I switched my research on the human level, at least, to being predominantly studying the opioids, because that's what we had in our collection. And at that time also, when we were studying molecular changes, or not the changes, but just trying to understand the molecular pattern of what's altered in the human heroin users, of course, we looked initially at what all, the neurotransmitter systems that everybody was looking at, as I showed you earlier, like dopamine. We also looked obviously at the opioid neuropeptide system itself. But one thing that was beginning to become evident as genetics started to become more prominent in addiction neuroscience was the high genetic load for opioid use disorder. And at that time, as people started to sequence and to understand the polymorphisms that are functional and are relevant to the opioid neuropeptide system, one of them that was very, well, at that time still wasn't very popular, but not popular, but it wasn't as well known, but it was the A118G polymorphism of the opioid receptor. And as I said, in the cell culture models, it had shown to be functional. So in looking at our population, we could see that indeed, shockingly, that a large percentage, in fact, nearly over 80% of the people in our postmortem collection of human heroin users, they had this A118G SNP. So, but what did it mean for the brain was, the key for us. And the thing that was important was, of all of the information that we had about people, meaning their demographic information, their age, their sex, and obviously, and so on, those factors actually didn't account for a large amount of the variability that we saw when we studied the brains, looking, for example, here at pointkephalin, but yet still, the polymorphism of the OPRM1 gene did. So, after that, we published a number of studies looking at genetic variants in regard to gene expression and protein levels in the brain that now, and then as genetics became, more popular, it was like impossible to publish anything that if you didn't have 1,000 people in your population, which for us is impossible. We don't have 1,000 people in our postmortem population. But fast forward, so what is it, 14 years later, and Joel Galenter from Yale and his colleagues showed that indeed what we had seen, so many years before, nearly 20 years before, that the OPRM1 polymorphism did account for meeting a GWAS significance and replicated, did account for the association to opioid use disorder. Another issue, as I said, in terms of trying to understand the vulnerability for individuals to develop a substance use disorder is not just genetics, but the genetics also impacts in relation to, like, for example, behavioral traits and psychiatric comorbidity. And a number of behavioral traits, again, this audience, you guys all know about, in aspects from, impulsivity, inhibitory control, reward sensitivity, and negative affect. And we could see, for example, taking individuals who had a cannabis use disorder and looking at polymorphisms that we could show had a significant relationship to the expression, for example, of proenkephalin or dopamine D2. Here's proenkephalin gene. That those that they also showed really strong relationship to anxiety, and we could look into the amygdala and see that the genetic risk there. But the thing that was interesting is that it's people who who were diagnosed with a cannabis use disorder, the greatest risk was, yes, if you had high anxiety, you had an increased risk, but even more if you had the genotype for this enkephalin. But it was a combination, that combination of both showing the high anxiety and high gene risk for here. This was the proenkephalin gene. So this synergistic contribution of negative affective traits and variants of proenkephalin, at least, were associated with cannabis use disorder. As I said, we could show a relationship directly in the human brain. But also aspect, as I said, anxiety is really key. Major depression has a high comorbidity with substance use disorder. Another area for me, as I said, in terms of understanding the neurobiology of it, if we looked at the amygdala, one of the things, again, coming back to the opioid system, was a prodynorphin gene expression was really fascinating. And it was fascinating, again, from the human brain perspective. When we looked at the human brain, there was a part of the amygdala that always showed this dysregulation, and in particular, in people that had a major depression disorder, but not those that had were diagnosed with schizophrenia. Interestingly, those changes that we saw in people with major depression disorder were matched in those people that we saw who were heroin users and even cocaine users. So what is that region? So this is the important thing for me in terms of, one, why I love the brain, because we still know so little. And in looking at the human brain, this dysregulation that we saw in dynorphin was specific to this region called the periamygdaloid cortex, the PAC. But the animal models did not study the PAC. Mainly, we've learned a lot about the central amygdala and the basolateral amygdala, but this we were seeing in an area that no one studied. So we were trying to figure out what it meant functionally. And so that helped us to go back to our animal models where we could have animals self-administer heroin and find the same region, the complementary region in the animal. And we were able to now develop a technique because we wanted to see what did it mean to the whole brain on a functional level to change the activity of these dynorphin cells in this really obscure nucleus of the amygdala. And so we combined dread technology where you can infuse a virus that changes the function, the G protein coupled activity of that cell, and that is only activated by an inert ligand, at that time at dose also clozapine and oxide, CNO. But we combined it with FTG micropath. So we can see what does it mean when you change the functional activity of that neuron in the, like I said, in that region of the amygdala no one had been studying, and see what it meant for the whole brain. And we called it dread-assisted metabolic mapping, or DREAM. And it was really, I mean, again, why I love the brain, it was amazing. Just changing the firing activity of those few cells recruited and activated the region called the extended amygdala. And the extended amygdala, George Hurd and Leonard Hemer, two amazing neuroanatomists, had identified this extended amygdala region as being, implicated, or should have roles, I should say, in maladaptive, for example, responses to fear, anxiety, and stress. So here we were being able to see that just these few cells could, we could also show it on a functional level, could contribute to not only recruiting this extended amygdala, but it could change corticosterone levels in the periphery, in the blood. We could make the animals anhedonic and change their depression-like behavior just by these cells. And that's what studying the human brain helped us to identify, because a lot of times and that's why, like I said, my love for the brain, my love, is based on the fact of this heterogeneity. Is the complexity is frustrating, but it's also beautiful, and it emphasizes that we still know, and we still have a long way to go to really learn a lot more about the individual cells and the circuits that they are part of that could even be targeted, perhaps for treatments for major depression disorder and addiction. Another thing that drove my, as I said, the complexity of addiction neuroscience and vulnerability comes back to aspects of neurodevelopment and trying to understand the developing brain. Again, complex, but I think, really important. And I was very lucky. Sometimes you go to, well, not sometimes, often you go to conferences and you speak to your colleagues, during the conference, you listen to a talk, you go to posters. I miss posters, especially posters, because you get to speak to the people, especially young people. But I was coming back from a conference on the flight, and I ended up sitting next to someone I didn't really know that well at that time, Diana Dow Edwards. And I was, at that time was studying more about cocaine. And I was saying I really wanted to look at understanding, we're seeing in the adult brain, what does it mean for the developing brain? And Diana came on a great journey with me in terms of trying to study the human fetal brain, because at that time in Brooklyn, where she was in SUNY downstate, they had a program, and we decided to say, okay, in terms of looking at aborted specimens and seeing whether or not I could study the molecular changes in the human fetal brain. So Diana was great. And the study, though, even though, as I said, we started off, at least for me, wanting to look at cocaine, what happened is, one, you have to get, obviously, ethical approval. You have to try and get grant money to fund the research. And that took a little while. And finally, when we got funding, some little funding to do the research, interestingly, the crack cocaine period, thank goodness, had ended. And there was the women who were using cocaine a lot while pregnant, that number had decreased. But an interesting thing was that the women had been using cannabis, pregnant women, and the numbers were still there for cannabis use during pregnancy. And so that's why most of our research focused then on developmental effects of cannabis from the fetal perspective. This is the adult brain where we can look at the cannabinoid receptor expression in the human brain. And just as everybody had noted from animal models, the cannabinoid receptor here, the CB1, is, a really highly abundant. It's the most abundant G protein coupled receptor in the brain. But in fetal development, this is the mid-gestational fetal period, we could see that it was expressed mainly like in these limbic areas like the ventral striatum, the nucleus accumbens, the amygdala, and the hippocampus. And we could see that in studying the brains of those fetuses whose mothers had smoked cannabis versus not, we could see significant changes in a number of systems. Here I'm just looking at, for example, microtubules that are important, for structural regulation, but even for genes that are classically associated with substance use disorders in adults, like the dopamine D2, which is, generally decreased often in adults with substance use disorder, and the debate whether it's the chicken or the egg, is it the drug that induces it or was it there before? It's a combination, obviously, of both. And here we could see that even during fetal development, exposure, you could see reduction of dopamine D2 in the fetuses, even at that stage. And in the amygdala, and we could even see during fetal development, gender differences, that it was driven in large part by the boys. When we look at the fetal, the human brain, we could use our animal model now to actually see what are the long-term effects, because that's the question for me, why I really wanted to, it's masochistic, but to look at the human, to see whether or not, one, like I said, we can replicate in the animal models where we can then follow, across time. And indeed, we could see that, prenatal exposure to THC generally led to increased self-administration here, for example, of heroin, even when under conditions where we said that they would have the same total amount of heroin, those adults that had fetal exposure to THC pressed the lever very quickly to get their first hit of heroin. But we also saw greater heroin self-administration even when we had exposure during the adolescent time period. And, this quote unquote debate about, gateway hypothesis of early exposure to drugs such as, THC or alcohol. But here we did definitely saw at least at that time. Now we know that there, even with animals, that there are behavioral traits that make a difference to that. But we could see in generally that there was an increase of heroin self-administration. Well, I forgot about this, that we were able to also go in, use laser capture, my video is off, and pick up and now start sequencing to really see what was altered in the cells in the adults with either prenatal or adolescent THC exposure. And one thing that it emphasized was aspects of epigenetics, that there are these really profound changes in epigenetic alterations. And, epigenetics, as many of you know, you inherit your DNA sequence from your parents in terms of the genetic contributions to gene expression patterns and so on. But the environment can impact on the control of that gene activity, not by changing the DNA sequence, but by changing how the DNA, whether it's in open or closed states, and these are epigenetic alterations, whether it's on the methylation of the DNA itself or acetylation or modifications, methylation of the histone tails itself. But these can be controlled by the environment. And so you can have changes that we saw in relation to epigenetic alterations in regard to, THC, developmental effects of THC, all came back to aspects of synaptic plasticity. So in large part, that these epigenetic changes that correlated to synaptic plasticity, that there was not, I should say, this great correlation to synaptic plasticity in normal animals. In adults that had, here is adolescent THC exposure, there's a complete reorganization of epigenetic and synaptic plasticity marks. But one of the things, as I said, is that perhaps my problem is that I ask too many questions, and we see these adult vulnerability with cannabis, and the question, though, is that, as I said, we see that it's epigenetic. And epigenetic, the original definition of epigenetics, is not really what we have today. Today, we talk about epigenetics in regard to the particular epigenetic mechanisms that can occur on the chromatin and so on. But the original definition of epigenetics by Conrad Waddington in the 1940s was really, it's not just the causal relationship between the gene and the environment, but it had to be inherited. It had to be assimilated in the genome in some way. And so because of this, I wanted to at least check to see whether or not there could be cross-generational transmission, if indeed what we were seeing was quote unquote epigenetic by the original definition, even if yes, on the molecular level, we did see these profound epigenetic changes. And indeed, when we look at next generations here, it's one of our first studies, we could see that adults had parental THC exposure, they self-administered more heroin, they had greater changes, electrophysiological changes in terms of glutamatergic transmission in the striatum. And we could even see it last even to their great-grandkids, that they had changes in, here, this is looking at food self-administration, was in terms of motivated behaviors that were different. So yes, there is something, and we're still looking into mechanisms, and we've recently identified a mechanism by which this could occur to cross, different generations. But so addiction, yes, complex disorder, and a lot of the, that we can see in subsequent generations. But an important aspect for me, and as I've now studied this for a while, is that, people, and I think that field of addiction and our society in general, we don't emphasize enough that a lot of the changes, even though we can see them long-term, because many are epigenetically driven, it's reversible. So the question is, can the neuroscience knowledge advance novel treatments? And so that's also, a huge part of the work that, I've done, because a big part of the path that I was on was not just to have direct, the research that I did was, I had hoped that would have direct impact in being able to understand the human brain, but also that, would it lead us to ideas about treatment potential. But we do know that, translation is very challenging. Most of the NIH basic science, the research awards, nearly about 4% of them are for translational research, and less than 1% of preclinical research gets really translated into human and into new treatments. So it's an uphill battle, but the fact is, and again, as all of there are only a very small percentage of people who suffer from substance use disorders really receive treatment. And a lot of that, we can talk about it in the question, the discussion period, comes back to stigma. Obviously, a lot of governmental regulation of the treatments, but at least for opiate use disorders, most of them are opioids themselves, with a lot of governmental regulation for the practitioners and clinicians to provide those medications for their patients. And we're still not advancing the science-based discoveries to treatments. So the question was, was, like I said, the naive me that went into this field. I really hope that the work that I did could lead us to that. And so, a lot of the molecular changes that we saw, not just in relation to cannabis, but actually heroin use disorder, we could see when we use unbiased strategies of sequencing, whether, even microarray to start and then RNA sequencing, led to the fact that there was profound changes once again in terms of epigenetics and related to synaptic plasticity, and where there's a great correlation here, for example, to glutamatergic impairments, that we could identify specific epigenetic changes in the human heron brain. It's not that all epigenetic modifications are altered, they're very specific. And in general, we could see that there were these changes in acetylation. And these acetylation that are put on the histone tails, they're read by these bromodomain readers. And the one important thing about these bromodomain readers is that they are a really strong focus of the cancer field, where they've developed many medications for them or targeted towards them. And so, these BRD inhibitors, the question for us was, can we start using that towards substance use disorder? And the first thing we, did our animal models. And at that time, the prototypical one, we could see that this BT inhibitor did indeed reduce heroin self-administration and seeking behavior, and when given systemically as well. The problem is the stigma that I never really appreciated till I started trying to move some of our research towards a medication aspect, is that a lot of pharmaceutical companies are very reluctant to have their medication investigated as a potential treatment for substance use disorder. And that was very shocking, still fighting. And once again, in an unbiased manner, when we looked across the epigenome, we could see that the locus that showed the greatest epigenetic change in human heroin users related to a gene that I should have studied but didn't until now, is a gene called FIN. And FIN regulates glutamatergic transmission. It sits at the postsynaptic density. And another thing that's really fascinating about FIN is that it hyperphosphorylates tau, or it phosphorylates tau, I should say, and hyperphosphorylated tau, as many of you know, is a pathological feature of a number of neurodegenerative disorders of the tauopathies, again, like Alzheimer's. And we indeed, when we had studied the human heroin use, the postmortem brains of heroin users, we'd actually seen years before with Eva Keller, my colleague I mentioned before, these increased phosphorylated tau in heroin users. And we were able to replicate that in our animal models that had self-administered heroin. But the thing that's really important, once again, there's medication that is an inhibitor of FIN that we could see in our animal models that reduced heroin self-administration. And the whole goal was now using the knowledge that we've gained about epigenetics and synaptic dysregulation in heroin users to bring all this to clinical trials. But once again, we ran into pharmaceutical companies reluctant to have their medication investigated in this regard. So that's a battle that we're fighting, just to be able to even do preliminary pilot studies in people with heroin use disorders based on some of the drugs that are already being developed. This one, the FIN inhibitor for Alzheimer's disease, and so, we'll see. But I'll end with one positive, at least, in the context of medication development. As I mentioned, a lot of the work that we did looking at the developmental effects of cannabis in our animal models was really using THC. But there are over 140 cannabinoids in the cannabis plant. And the goal was, to at least look at another cannabinoid. And at that time, I said, okay, let's just look at CBD, because CBD was the second most, the highest cannabinoid in the cannabis plant. Today, the concentrations are much lower in the plants that are used recreationally. But in looking, and so there's this yin yang with THC and CBD that we've seen and many researchers have seen. And here, as I mentioned before, when we normally gave THC, animals increase their heroin self-administration. But when we gave CBD, we actually saw an interesting phenotype. It decreased heroin-seeking behavior, and specifically heroin-seeking behavior that was associated with environmental cues, like when the animal self-administered heroin, a light would go on in their environment, and if we showed them the light later without them getting the drug, they would press the lever a lot. And so this is what CBD decreased. It also normalize some of the glutamatergic or the synaptic the genes and proteins related to glutamatergic synaptic function. For example, the GLUA1 receptor that's normally dysregulated in heroin users, animal models at least, CBD normalized. So at that time, cannabidiol was not very prominent as it is now today, but I wanted to still check to see whether or not it may be a potential treatment for opiate craving, at least. And even though a pharmaceutical company at the time was reluctant to have their medication investigated for addiction treatment, they were open to getting more clinical trials with their product at that time because it was, new. And here we were able to see that, similar to our animal model, that people who are shown a heroin cue and had received placebo, they craved, and CBD reduced that. We could also see that what we hadn't studied in the animals at that time, but now we've replicated in our animal models, is that it actually CBD reduced anxiety that's triggered by the heroin cues as well. Another reason why I had thought that CBD might be interesting to develop as a potential treatment for heroin use disorder, especially in relation to craving, was because we saw in our animal models that weeks after the final CBD, it still had an effect. And we saw that in the human subjects as well, that even 7 days after the last CBD administration, it still reduced craving and still reduced anxiety. And it also reduced the cue-induced effects physiologically. So it's not just self-reports, but we saw changes in regard to reducing, normalizing cortisol and heart rate. But as I said, now CBD is everywhere, and and becoming, unfortunately, mainstream to a manner that people think that it's like a cure-all, and that it's in your coffee and your hamburger and so on. But it's still something that for us that we're, trying to develop, and we're carrying out large clinical trials right now with it. And this is people, even for the dogs, people everywhere I go, people tell me that CBD helps their dogs. So for me, the road to becoming an addiction neuroscientist took many, twists and turns from Jamaica, but I think the foundation was there in terms of, just like I said, the curiosity, the interest in the brain, which was a little weird for a little girl. I have no idea where that came from, but it's been an interesting ride. And as I said, I started off with my mom, and I'm going to end with my mom. One of the things about studying the FIN inhibitor was that, as I said, it was being developed for neurodegenerative disorder, and my mom unfortunately had Alzheimer's. And part of me hoped that something I could give back to helping, understand that, and developing treatments as well to help her. But unfortunately, she passed away. But one thing my mom, sorry, I'm getting a little trouble. But one thing my mom did, I think, the foundation is character. And one of the things for me, both from my mom, who was my hero, and my science hero, Albert Einstein, I think a lot of people, when the path to neuroscience or any science is like, they talk about, as Albert Einstein said, many people say it's the intellect which makes a great scientist. They're wrong. It is character. And so true. So my mom taught me, and indeed, she had her Einstein face once in a while as well. So I'm going to end there because I know we have probably a lot of questions, but I want to frame part of the things for me as I was going through this in terms of understanding addiction, love for neuroscience, the addiction of addiction neuroscience. And as the director of the Addiction Institute, one of the things I didn't talk about, and that is a really strong foundation for me, is that I get letters and calls all the time from families affected by substance use disorder. So yes, addiction is about the individual, but it's the impact is so. But from our field, I do think that academic reality for addiction really differs from addiction reality, and a huge part of it is about the urgency and what we choose to nitpick on that may not really come back to helping that individuals, the families, and the communities. So with that, I will end, and so we can talk about anything that I've mentioned, and hopefully continue our discussion in the love of addiction neuroscience. Thank you.
Thank you so much, Yasmin. That was so inspirational and so
Thank
real.
you.
And I applaud you for being so persistent against all odds. I'm sure your journey was not as simple as you perhaps portrayed it there. I'm sure it had bumps and it had dead ends and, perhaps even potentially run over by a car as you're going through your journey.
Yeah, exactly.
And that leads me to a question is, what is the message that you would give to the younger generation? They might have the character, they might have the rest, but what are the components that you think would allow them to go and survive and prosper along that journey, which is a long journey?
It's a long journey, but I think the thing that's important is to break up your journey. I definitely had this vision of what I wanted, and I think it is important to have a vision of what you want to do. Because I will tell you, a lot of people along the way told me I couldn't do this or couldn't do that. But because I knew I wanted to study the human brain first, for example, something like that, I kept finding the right mentors. So the question is, as you are going through your journey and early on, and not just early on, I think every stage of your career, you need mentors and, people who believe in you. And they don't necessarily have to be scientists themselves. They can be friends. They can be people from other fields. But I think without having a good mentor, team, I think that sometimes the negatives that you can encounter along the journey can kill your passion, kill your curiosity, and even though, like I said, you have to persevere, you can get beaten down. But I do think that it's the environment that you put yourself in, and being around positive people. When you're surrounded by people who are always negative, you're going to start believing that. And I really fully believe that, like I said, your team, think like you're building your own team. Your team doesn't, yes, there should be scientists, but doesn't have to only consist of scientists. I think that, people from different walks of your life, even your peers can be part of your team because you're growing together. Remember that, a lot of people who you are, your fellow students and people who, fellow peers who you meet at conferences, you're growing in your career with them. And they're great. I was very lucky to meet a number of women, because in the beginning there were not that many women in some of the neuroscience conferences, and it was great, having little sisters, and feeling a part of a community. So that, I think, really helped me.
Thank you, Yasmin.
Okay, if I may just add, because it's a case that's quite unhappy now, in the sense that I and my fellow colleagues know these two people, both of them, but they do not know that we know both of them. They do not know that each other is in treatment in the same treatment unit. And the daughter has been diagnosed, the mother has been diagnosed with paranoid schizophrenia and is well in treatment with antipsychotics and is doing well. And the daughter keeps being psychotic, and the hospital keeps saying this is just psychosis due to drug use. Right. And we keep saying this is hereditary, mom has got chronic paranoid psychosis, but we cannot say this aloud because it cannot be written in a patient record, because the daughter is not supposed to know that the mother is chronic paranoid psychosis, because she's not supposed to know it. So it's really difficult. I got this knowledge and I cannot get it to a psychiatric record because she's not supposed to know that I know them both. So it's really ethically difficult.
That's challenging. That's very challenging. And that is one of the things, obviously the patient confidentiality is critical. But one of the things that we have for substance use disorders, at least, I know like in the US, for example, your other physicians who treat you for other things don't have access to your substance use history. So that I had, know people who were going to have, their cardiologists, because they have major heart problems, did not know that they had a cocaine use disorder. And how do you treat someone and give them the best medical care when you don't have a full assessment, because aspects of psychiatric and addiction those diagnoses are considered still the stigma. So it's the stigma that is preventing, I think better, as I said, better care. But yeah, I mean, clearly it's a genetic issue, the psychosis, and so many studies have shown the early, psychosis, the early vulnerability for substance use risk. It's challenging. You have a very tough challenge.
Yeah, I can't solve it right now, but I'm trying to find a psychiatric consultant who will actually listen, because I know I've seen mother and daughter, they're very similar, unfortunately. Yeah. Okay. Yeah. But thanks. Yeah.
Persevere, Irene. Persevere. Do things in spite of. That's the mantra. Do things in spite of. And we have another question from a colleague from Mexico, Sylvia Cruz. Thank you for excellent presentation. Could you say something about CPD drug interactions?
So the CBD, like every other drug, can interact with other medications. I mean, for example, with benzodiazepines, it is a biologically active compound. So many people think, I won't use the word benign, but it's organic, it's from the plant, but absolutely it gets metabolized obviously by the CYP, your liver enzymes, and so any medication that has a similar target will, as I said, aspects of benzodiazepines and so on will also be affected. So there's a long list of potential medications that CBD can interact with.
Thank you. And if you don't mind, we'll move towards stigma. I think this is something should not be ignored. And I remember in one of your slides, stigma kills. And I think today or yesterday, there was the information coming out from the US around 90,000 overdose deaths, has increased from 70,000. And the hidden message is that this is not necessarily about COVID. It's more about stigma, and that stigma kills. And before I ask you your opinion on that, which I think is a very pertinent one, we have a question here from Mehdi Faroukna. Thank you, Dr. Hurd, for the great talk, relevant to your discussion around stigma, as well the disconnect between scientists and the community. I was wondering what your thoughts are on recruiting people with lived experience of addiction into the field. I am not aware of any initiative in that regard, but seems like a missed opportunity.
That's a great idea. And I mean, I do know, so in the clinical enterprise of treating substance use disorder, there are a lot of people who have the lived experiences. A lot of the counselors, the social workers are people who've had that and have been very helpful and beneficial to helping patients, in that regard. There, of course, scientists and physicians, they're human, and there are people who have definitely had a substance use disorder, but because of stigma, they're not going to necessarily come out and say, I had suffered from this or from that. People might tell you in private, and of course, you're not going to, divulge it publicly. But there is something to what the idea that you have. I do think that especially for young people who are in communities who are particularly, inundated with the damage that substance use disorder, the peripheral damage that it causes, I think we should target them for getting them involved in the neuroscience of addiction. I think that's a great idea. I think that the more knowledge people have, and especially when it's close to them, they will ask questions that we're not asking. So like I said, my friends were correct. My friends were like, what, you're going to go and become a drug addiction researcher? You've not taken drugs. Yeah. There is something to be said if you've, walked the path or known people who walk the path, they will ask a different science question. And I think that sometimes that's why we're not advancing, like I said, the urgency of the questions that we might be asking. So I love that idea. That's a really great idea.
And that reminds me, Yasmin, of, we all carry our own cultural bias, and there are certain research that we do, and that is that we don't even think we should do. And I'm going to ask you a question which is very pertinent in the UK, I'm sure in other countries, is what are your thoughts around psychedelic research that is happening, you know, in parallel with cannabinoids and similar substances?
Yeah, I'm torn. And I'm torn because I don't want the public to think that these substances are benign and look, you know, now magic mushrooms once again, you know, will be a cure-all. Because I have people telling me that, you know, mushrooms can now cure psychosis and cure addiction. We need the research. And so I think it is important to do the research, but I don't know if it happened in Europe, but in the US, we had, you know, these drive-by opioid clinics where people are like, oh, we can treat your pain, just drive-by pass. And now they're starting to have these drive-by psilocybin psychedelic clinics. And people don't understand is that the research that has been done with the psychedelics, it's a process. It's like you know, you're there for hours. The therapist walks you through while you're, you know, that you have consumed the psychedelics, but under controlled setting. It's not that they're giving it to you and you go home and take this. It's a very structured treatment program. And so I'm afraid that once again, the public and people who, you know, might want to have an excuse perhaps to use, think that this is just, they can just go and just buy from whomever. And especially today on the internet, you can get anything. So I am torn. I think that we have to be as scientists very clear to the public that this is not your normal, you know, magic mushroom. This is not, there is a clinical procedure with a clinician that you have to go through in order for at least what the research shows that so far. And it might turn out to be the psychotherapy that is doing the most benefit, not the, you know, the psychedelics. So, but you need research to really answer that.
Thank you. I think that means a lot to a lot of us, you know, hearing it from yourself, Yasmin. Last but not least, I know Jason Winter has put into the chat. He's trying to create an attention because he comes from Jamaica, and I think he's saying hello to you. But he mentioned here, and perhaps Jason can explain to us, we have a group of graduate students from Northern Caribbean University in Jamaica turned in. I'm not really sure what that means. Perhaps it's a Jamaican term, turning in.
Well,
I'm not sure.
Hello, good to be, you know, following the series. I've been with you a couple months. I think I missed the last 2 months, but I'm so happy that I didn't miss this one. And when Yasmin started in our introduction to acknowledge her roots, I said, yes, this is the one that, you know, God made me ensure that I don't miss it. So it was just good sitting in. Northern Caribbean University is an Adventist institution. We do have a graduate program, counseling psychology. Both undergrad and graduate, the students do take a course in substance use, looking at the impact, you know, on behavior and how we can plan interventions at the different levels. It doesn't take the hardcore physiological or neuropsychological angle or twist that has been advocated throughout the series here, but it's so refreshing to get that angle of it because, you know, human behavior does have a large underpinning in the science and the neuroscience of it. So it's good to be here. I know we had Leighton Campbell. He was on, might have slipped off, but good to have listened to you and to be following the series here from Jamaica.
Great, Jason. Really proud and happy to hear that you are all, you know, following this series because I think it is important. You know, the great thing about science, I forgot to really say, it's embodied here. It's international. And so that's the thing, you belong to a very special group, you know, and it's really great to have that international tie. You belong to an international family. And that's what I love about science. There are no borders. So I'm really glad that you guys are part of it.
So I you know, the International Society of Addiction Medicine president is here. So I may ask Dr. Alex Baldacchino to maybe explain a little bit about the conference.
Thank you. Panil is a great example. She's an amazing ISAM ambassador. to supporting the values of the International Society of Addiction Medicine. But essentially, we try very hard to connect, and as Yasmin said, science, patients help us to connect, to support each other, and that's the main function of the International Society of Addiction Medicine. We do have our annual Congress. This year is going to be virtual, similar to last year, and you know why, and I'm not going to mention why, because everyone knows why, and it's very sick to hear the word again. But hopefully next year we'll be organizing our annual Congress in Malta, and the year after in Marrakesh. But this year, the call for abstracts has been open and extended to August 15th. There are about 6 domains. One of them is neuroscience and addiction mechanisms, but there are others such as stigma and inequalities, practice policy, related topics, and of course, clinically relevant guidelines and discussions around how to help and support people through a biopsychosocial program of activities. So please do not feel that this is a conference just for medics. It is for clinical scientists, non-scientists, undergraduate, postgraduate students. We have opened the membership to all of the above, and including pharmacists and non-medical clinicians such as nurses and occupational health professionals. So please do go to the webpage here, and also do go to the revised ISAM webpage, where you would see all other activities organized by our 5 interest groups, including neuroscience interest group. So we look forward to seeing you in abstract form in September, when we're going to start this second series of the In Love with Neuroscience, but also again virtual form in November. And last but not least, can I thank Hamed Ekhtiyari, who has been inspirational, and unfortunately couldn't join us today because he's going through his own journey, and he's moved from one job to another. But he is a great example, and as you have heard him talk in our first talk of the series, he has been an amazing person, not just for his colleagues in Iran and the US, but for a lot of people in the globally. And I'm glad to say that Hamed has been appointed as the co-chair for the ISAM Regional Council, together with Professor Gregory Bunt. And I'm sure that he will do a grand job there as well. Thank you very much. And keep safe.
Thank you. Keep safe. And I'll make sure that you receive the follow-up email comprising all the videos of the 12 talks that we had, and also the information regarding the next round of talk series. Take care.
Thank you.
Okay.